So in this video we are gonna talk about the blood physiology. This is our first lecture on blood physiology and the first very important topic, a high yield topic, the pretty much pretty likely to appear in your BCQs, your MCQs, and other portions of your exam. So now let's get started. Okay, so blood composition. First of all, let's look at the definition of blood, what blood actually means. Okay, so blood. This is the definition of blood. Blood is a specialized connective tissue. Okay, this connective tissue is a tissue type of the body. Tissue, as you guys know, that is a collection of cells which are specialized to perform a particular function. So a group of cells. So it is a specialized connective tissue of the body which is composed of a solid portion and a liquid portion. The solid portion includes your blood cells and platelets, and the liquid portion includes your plasma. So it is composed of platelets, blood cells, and plasma, and its volume is approximately 5 to 6 liters. So 5 liters approximately of the blood of the total blood volume in the body is 5 liters, approximately making almost 8%, 8 to 9% of your body weight. Okay, now so its viscosity is actually the resistance to flow. So since it is blood overall is a liquid, so it flows from one point to another, right? So the friction or the resistance to flow is known as viscosity of a liquid, and in this case viscosity of blood. Viscosity of blood is 3 times as the viscosity of water, so it is 3 times as viscous as water, and the viscosity is mainly due to the blood corpuscles which are present in the blood. So the greater the blood corpuscles, the greater the viscosity. Now let's look at the basic components of the blood. I've made a chart so that you guys could easily remember it. Okay, this is your whole blood in this black circle over here. The whole blood is divided into 2 types as we talked about. It is divided into the plasma portion, which is the fluid portion of the blood. It is 55% of the blood is your plasma. So 55%, more than half of the blood is liquid in fluid form. So that's why it appears to flow from point to point, and the liquid properties are more dominant. And 40 to 45%, the remaining 40 to 45% of the blood is composed of blood corpuscles and the formed or the form elements. Formed elements of blood corpuscles are the same, are almost the same thing. Okay, they can be used synonymously. Now, blood plasma and blood corpuscles are further divided into subdivisions. Plasma is made up of 2 things. It is made up of water, mainly water, because plasma is the fluid portion, so contains predominantly water. The 90% of plasma is water, and the remaining 10% of water is made up of solid portions, the solutes, dissolved solutes, which include amino acids, your nitrogenous waste products, your nutrients, example glucose, amino acids, other nutrients which are present in the blood for transportation purposes, so they are dissolved in the plasma. Electrolytes, which are very important to carry out certain functions of the body, so example the sodium chloride, calcium bicarbonate, and other many electrolytes which are present are present in the dissolved form in the plasma. It may also contain dissolved gases. The main gases that are present in the plasma include your carbon dioxide and nitrogen. Okay, these are the 2 main gases present in the plasma. And plasma proteins are also present. The plasma proteins include albumins, globulins, and fibrinogen. Albumins and globulins act as transport proteins, whereas fibrinogen has a major role in blood clotting. Albumin is the most abundant plasma protein, and it is the due to the proteins present in the blood, since they are again solutes of the blood, they exert some osmotic pressure on the liquid present inside the blood. So, and due to this osmotic pressure, the liquid stays within the blood capillaries and does not easily leak out of the blood capillaries. So, and since they are proteins, so the osmotic pressure caused by the proteins on the fluid portion of plasma is known as oncotic pressure. It has a specialized name. A special name is given to the osmotic pressure due to the plasma proteins. It's called oncotic pressure. And the major contributor to the oncotic pressure is the most abundant plasma protein, which is our albumin. Then uh, okay, we've done this. Most abundant plasma protein, 55% of the plasma protein includes albumins. Okay, and I've made a capillary diagram over here. These consider these. Just imagine these two branches as two branches of capillaries, and the space in between the branches shown by these green colored circles. Consider this space as interstitial space. So at the arterial end of the capillaries, the fluid portion of the blood, the plasma, most of the plasma moves out of the capillaries into the interstitial spaces due to the down the hydrostatic pressure. It's due to the hydrostatic pressure at the arterial end being higher than the hydrostatic pressure in the interstitial spaces. So this causes the blood fluid to move out, but it does not due it moves out due to the hydrostatic pressure applied by the pressure difference in the blood plasma and the interstitial fluid. Now, in the reverse direction to the hydrostatic pressure, another pressure that we just spoke about, the oncotic pressure acts, as shown by these blue arrows. So the oncotic pressure prevents the fluid. The plasma proteins are present in this plasma. They cannot escape the capillaries, as you know, because the plasma proteins are quite big in size, so they cannot squeeze through the pores of the blood capillaries, because blood capillary pores are quite smaller than the plasma proteins. Now, what these plasma proteins do is that they apply an opposing pressure against the hydrostatic pressure, keeping the blood keeping the blood fluid within the blood capillaries. Okay, so this is the role of oncotic pressure, which is due to the presence of plasma proteins. Major contributor is albumin, which is 55% of the plasma proteins. Albumins also act as transport proteins for certain lipids that cannot dissolve in the aqueous medium of the blood. Since lipids are hydrophobic, they cannot dissolve in the blood. And so if they cannot dissolve in the blood, that would simply mean that they could not be transported in the blood. But if we have transport proteins like albumins and globulins, both of them act as transport proteins for lipids and steroids. So the steroids will dissolve in them, and so they'll easily transport the steroids and the lipid components across the blood to different portions of the body where they are needed. Fibrinogen, as I spoke about, has a major role in blood clotting. I've listed a few another role of globulins as well. Well, globulins are mainly made by the liver, and another function is that they act as antibodies. Some of the antibodies are entirely made up of globulins. They are known as immunoglobulins. Immuno because they provide immunity, globulins because they are actual globulins of the plasma. Immunoglobulin A, IgA, abbreviated as IgA. They act as antibodies against certain infectious agents. Okay, they also act as transport proteins. We discussed this. Okay, certain ions can also be transported with the help of globulins and lipid transporters. We spoke about the lipid transportation just a few minutes earlier. Okay, okay, we talked about the plasma proteins. Then there is a term for that portion of the plasma which only includes your albumins, your globulins, but lacks fibrinogen but does not contain fibrinogen. That portion of plasma is known as serum. Since it lacks fibrinogen, so serum cannot coagulate. It this serum cannot cannot clot, cannot coagulate. Okay, so it has no role in blood coagulation, in repair of damaged arteries, damaged veins, damaged blood vessels. Serum is a yellowish part of the plasma. It is part of plasma, okay, that does not contain fibrinogen. It lacks the blood clotting plasma protein and only contains albumins and globulins. Albumins and globulins are the transport proteins, the proteins responsible due to all the proteins. Oncotic pressure is present. They are the transport proteins, and globulins act as immunoglobulins. Albumins are the most abundant plasma proteins, in the plasma. So part of the plasma that does not contain fibrinogen and only contains albumins and globulins, and is yellowish in appearance, and that does not clot is known as serum. Okay, so these are a few properties of serum that I've listed. That it does not clot because it lacks fibrinogen, and it's yellowish in appearance and contains mainly albumins and globulins, and it's plasma. So this type of plasma is known as serum. Okay, so we've talked about serum, we've talked about plasma proteins, we've talked about the composition of blood plasma, and it mainly contains water. 90% of blood plasma is water. Okay, we've talked about oncotic pressure. Now, let's talk about the other half of the blood, which most of our discussion in the upcoming videos is going to be on the other half of the blood, your blood corpuscles. Okay, blood corpuscles, which are also known as the formed elements, they make up 40 to 45% of the total blood volume. Okay, three main types of blood corpuscles are present in the blood. The first type is known as platelets. Platelets. The second type is known as leukocytes, which are actually your white blood cells. Another name for white blood cells is leukocytes. They mainly provide immunity. Platelets are for clotting purposes, clotting, repair of the damaged blood vessels, etc. And erythrocytes are the third form, the red blood cells. Erythrocytes is another name given to red blood cells. These are the most abundant cells of the body, not just of the blood. They are the most abundant cells in the blood, but they are also the most abundant cells throughout your body. So no single cell type is greater than the number of the erythrocytes. They are 25 trillion in total, and we've got a detailed explanation in the next video on erythrocytes. The link is given in the description, so check that out after this video. All right, now after let's talk about them one by one. Platelets, they are pieces of large blood cells. They are not cells. Make this clear, guys. Platelets are not cells. Okay, they are not entire cells, not cells. They are pieces of big cells. Okay, there was a big cell, megakaryocyte. Megakaryocyte was its name, derived from the bone marrow. It broke up into pieces. These pieces are now called platelets. They have a very essential role in blood clotting. So if any artery or vein gets damaged, so the platelets are necessary in addition to the fibrinogen and in addition to the other clotting factors to clot that damage and to repair the damaged blood vessel. Okay, so this is all about platelets that we are going to talk right now. We will go in a bit detail in the upcoming lectures. Now the next type is leukocytes. Again, we have a detailed lecture on leukocytes in the immunity section. Okay, but I'll talk about its subdivisions. There are two major subdivisions of leukocytes. We have granulocytes and agranulocytes. These are called granulocytes because they contain granules. Granules contain certain chemicals, certain substances. They can be histamine, they can be heparin, they can be other digestive hydrolytic enzymes, etc., which are present in their cytoplasm, and they contain numerous fluid-filled granules in their cytoplasm. Okay, and the examples of granulocytes include neutrophils. Neutrophils are also the most abundant white blood cells. Most abundant white blood cells, WBCs, they are 60% of the WBCs, okay, not of the blood. 60% of the WBCs are your neutrophils. They are granulocytes and present in the blood plasma. Uh, yeah, blood plasma. Then comes basophils. Basophils are also leukocytes. They contain certain granules as well. Granulocytes, basophils, and then eosinophils. Basophils and eosinophils. Okay, basophils also have certain roles in immunity. Eosinophils also have certain roles in immunity. We will talk about them in detail in the upcoming lectures. Okay, now let's talk about agranulocytes. Agranulocytes are those that lack granules in the cytoplasm. These are your all the lymphocytes, the B and T lymphocytes, lymphocytes, the B types and the T types. And the second type includes monocytes. Monocytes are monocytes give rise to macrophages once they they filter through your blood into the surrounding tissues. In the surrounding tissues, these monocytes give rise to macrophages, and then these macrophages are long living, long lasting cells. They have both monocytes, macrophages, and neutrophils. All these three have phagocytic activity. Neutrophils, monocytes and macrophages have phagocytic activity. This means that they can phagocytose foreign particles and then digest it inside of them. Okay, so this was about the classification of leukocytes, white blood cells, and then the third and the most abundant type of the blood cell in the blood, the erythrocytes. So the red blood cells, they lack nucleus in matured form. Well, in the immature form, when they are newly made, they do contain nucleus, they do contain mitochondria, and they do contain endoplasmic reticulum. But as they mature, they lack nuclei, they lack mitochondria, they lack endoplasmic reticulum, so as to provide more space for the hemoglobin. Hemoglobin is a protein, a pigmented protein present inside the erythrocyte, which is specialized for combination with oxygen. It contains iron 2 positive ions in combination with the tetrapyrrole ring. The tetrapyrrole ring and iron collectively form the heme group, and then that heme group transports oxygen to other portions of the body. So the hemoglobin, so as to provide greater room for the hemoglobin inside the erythrocytes, the nuclei, the mitochondria, endoplasmic reticulum are removed, are digested as the nucleus, as the erythrocyte, uh, as the erythrocyte matures. Its average lifespan is 120 days. So after 120 days, the erythrocytes are broken up. They die. They self-destruct in the spleen portion of the body. In the spleen, they become fragile, and then they at the spleen through the spleen pulp. At the spleen pulp, the fragile cell membranes of erythrocytes, they rupture, and the erythrocytes, they just self-destruct. They die out. Okay, after 120 days. We have a detailed lecture on erythrocytes in the upcoming lecture, upcoming videos. So the link is given in the description. Do check that out, okay, immediately after this lecture. It's important so that you could link the two lectures together. Approximately 25 trillion cells in the body. Erythrocytes are 25 trillion in total in the entire human body. The 5 liters of the blood collectively contain 25 trillion erythrocytes. That's a very large number. Okay. Now, another term related to erythrocyte is hematocrit. Okay, if we to sum up the volume of red blood cells, the total volume of red blood cells, and compare it and take the ratio of that volume to the total volume of the blood, that ratio that we get is known as hematocrit. Or in other words, we could simply say that the volume percentage of red blood cells in the blood is called hematocrit. What does it mean? Okay, hematocrit is also called the packed cell volume. Okay, why is it called the packed cell volume? Why do we call it the percentage? Okay, now the formula for calculating the hematocrit would be the total volume of red blood cells. All the red blood cells which are present in the blood, just sum up their volume divided by the total volume of the blood. This ratio can be called hematocrit, or you could just multiply it by 100 to express it as a percentage. Okay, this ratio is known as hematocrit. The cumulative volume hematocrit. A total volume of red blood cells compared to the total volume of the blood is hematocrit. So the percentage volume of red blood cells in the blood is called hematocrit, and the normal values are 40%, 40% in females, 45% in males. Okay, and now after this, these are the values that you should know, you should learn. Okay, these are important with respect to your papers, your exams. Now let's move on forward. Okay, the plasma portion, the hematocrit, and other blood corpuscles, they can all be separated by a technique known as centrifugation. The centrifugation technique is a very good technique that separates all the three components of the blood. Okay, in this technique, a blood sample is taken and in a test tube, and then it is spun. The sample spins at a very high speed, uh, and that all that spinning, what it does is that it causes the heavier particles, the heavier portion of the blood to settle at the base. The heavier portion of the blood, which is mainly your erythrocytes are the heaviest portion of the blood. They settle at the base, and this base volume over here, as I told you guys earlier, this would represent your hematocrit. So the percentage of the red blood cell volume present in the blood is your hematocrit. So if we express this in as a percentage of the total blood volume, this will represent your hematocrit. And then the plasma, as I told you guys earlier, is the fluid portion of the blood. It will stay on the upper side, because it's the fluid portion, it's the lighter portion. It will stay on the top surface, and in between the plasma and the hematocrit, in between the plasma and the hematocrit, we'll get a ring which is known as buffy coat. Okay, which is known as buffy coat, and it is mainly composed of leukocytes, your white blood cells, and platelets. Okay, the other two components of the blood. So because their quantity is very little as compared to the plasma and the hematocrit, so they'll come over here as a thin ring in between the plasma and the hematocrit. And this separation is mainly on the basis of their density, on the basis of their weight. The heavier ones go on the at the base, and the lighter ones stay on top. Okay, this is a result of centrifugation. Erythrocytes, 45% of the total blood volume. This is your hematocrit value is given over here. This is a hematocrit value of a male, a normal male. And 55% of the total blood volume is your plasma. As I told you that plasma ranges between 55 to 60% of the total blood volume. Okay, this was centrifugation. Now after this, I have a few images of leukocytes that I want to show you guys. Okay, right over here. Okay, these are your monocytes. If you look closely, the cytoplasm is clear. The cytoplasm does not contain any granules. This is eosinophils. The nucleus has two lobes. It is a headphone shaped nucleus, okay, a bilobed nucleus, and contains several granules. Granules. This is called eosinophil because it is stained by the stain eosin. Okay, stain eosin is taken up by these granules, uh, and these granules give an eosinophilic appearance. Okay, eosinophilic because they take up the eosin stain. Then basophils also contain granules, so both of them are granulocytes, and we've talked about the granulocytes up there. Okay, these are also granulocytes because they contain granules. Then we have our lymphocytes. Lymphocytes are agranulocytes, monocytes are agranulocytes. These are your lymphocytes, and this is a multi-lobe nucleus, the neutrophil. Neutrophil is a granulocyte because it contains several granules in its cytoplasm. Okay, so neutrophil, lymphocyte, monocyte, eosinophil, basophils, etc. Okay, granulocytes, agranulocytes. These are two composition, the five types of white blood cells present in the blood, the five types of leukocytes in the blood. Okay, so we've talked about the leukocytes, we've talked about the plasma. Okay, so pretty much everything is covered. Now the functions of the blood is left. Let's cover this one as well in this lecture, and then we are done for this lecture. Okay, functions of the blood. Okay, these are just a few functions that I've listed. There are many more, and simply something that you guys need to learn. Okay, let's go over them one by one. Okay, transport of cells. Okay, the first and the primary function of blood is transport. It acts to transport different substances. It can transport the blood cells itself, the white blood cells for immunity purposes, the red blood cells to transport oxygen, and other cells in the body which needs to be transported through the blood are transported by the blood. Hormones can be transported to the site of action. Enzymes to the site of action can be transported. Antibodies to the site of action can be transported by the blood, and other related substances. Certain waste products can be transported by the blood. For example, the urea, the carbon dioxide, all the metabolic waste products can be transported by the blood to their respective excretory organs. to be excreted. The second important role of blood is that it regulates pH. It regulates pH due to the presence of certain buffers. Buffer is any solution that resists changes to its pH when small amounts of acids or alkalis are added to it. So if we add an acidic or a basic solution to the blood, then its pH will not vary significantly due to the buffer system present inside the blood. There are several types of buffers present in the blood. One type is your bicarbonate buffer system. Okay, then there are the electrolytes of the interstitial fluids. They can also be transported by the blood and they can be regulated by the blood. So if the interstitial fluids they lack the electrolytes, they have a lower concentration of electrolytes, so electrolytes can add up from the blood to the interstitial fluids. Or if they have excess electrolytes, then the electrolytes can diffuse from the interstitial fluids back to the blood, thus regulating your electrolyte balance of the interstitial fluids. Then the third important function is that it provides defense against pathogens and other foreign particles, toxins. Okay, pathogens due to leukocytes, the white blood cells, the immunity that they provide, and the antitoxins to neutralize them. Then absorption and distribution of heat across the body. Okay, this is again a homeostatic function of the sites where the heat is generated. For example, your liver generates most of your heat. It is absorbed by the blood and then transported throughout your body to maintain and distributed across your body to maintain a uniform temperature across the body. So again, helps in homeostasis. Homeostasis, by definition, is the maintenance of a relatively constant internal environment. So this is again an example of maintenance of a constant internal environment, maintenance of constant heat throughout the internal environment of the body. It is done by the blood. And then the last function that I've listed over here is that it limits the blood loss through the damaged blood vessels by providing essential clotting factors, plasma proteins, platelets for clotting. All right. So if we have certain hemorrhage, for example, hemorrhage has occurred. Hemorrhage is the damage to blood vessels which leads to the loss of blood from the blood vessels into the interstitial spaces. Due to hemorrhage, uh, the blood vessels have been damaged. So if the clotting doesn't occur and the damage is not blocked, then it might lead to severe consequences. It might lead to extra blood loss from the body and can lead to very dangerous conditions. The it the blood prevents any extra blood from being lost by by repairing the damaged blood vessels, by clotting by the different clotting factors and the plasma proteins, the fibrinogen which we talked about up there, and the clotting factor and the platelets which we've already discussed about. If all these three are present in the blood, then they'll help repair the clot. Okay, so these were a few functions of the blood that I've listed. Okay, so that's it for this lecture in this video. Okay, the next lecture is very important, guys. It's on erythrocytes and a very detailed one. So please do check it out. Thank you so much.